going to start with Aurelie Bussy, who is going to talk about PCA, followed by Elisa Guma, who's going to talk about PLS, Lani Cupo, who's going to talk about canonical correlations analysis, and Raihaan Patel, who's going to talk about non-negative matrix factorization. We'll try to take a little bit of a different organization. So normally the talk is a little more conversational, but we're a bit pressed for time based on our rehearsal talk. And it would be great if you could just maybe back end your questions for each of the 4 presentations, if that's all right. Or if you have questions and you don't want to forget them, put them in the chat, and I promise to go through and go back and scan the chat afterwards. But let's try to let each of these individuals finish their talk to completion. And I want to thank them personally because they put a lot of work and time into getting these talks ready for this kind of more educational series. So just to motivate what we're going to be doing today, I think, one thing that is very prevalent in the neuroimaging literature these days is the use of several multivariate methods. And a lot of that has to do with the way in which we're starting to look at neuroimaging data. So importantly, what ends up happening is that one, all neuroimaging studies tend to have several different phenotypes that they can examine. So several different phenotypes could be functional traces from the bold signal at different regions of interest across the whole brain. They could be having structural data, functional data, diffusion data, all in the same individuals. And then within each individual, you have a number of different behavioral metrics, which you see here in this matrix at the bottom. And each dot represents kind of someone being positive on some sort of, say, neuropsychiatric measure assessed in a specific individual. And the question we now have is, instead of, you could do something quite simple, which is take, for example, one functional trace, which would be one column in this matrix, for example, and ask the question, how does that relate to any one of these behaviors? But we know a lot more than we did a couple of years ago. A lot of the functional traces, for example, or a lot of neuroimaging phenotypes have a high correlation or covariant structure within them. So if you're high on one or low on one, you might be high or low on the others. And same thing goes for psychiatric disorder, psychiatric symptomatology, and they may cluster together in different domains. And so I'm stealing this slide from Ted Satterthwaite's group. This is a slide probably shown by Cedric Zia when he gave a talk here in our series, and he demonstrated a beautiful method called canonical correlations analysis, which helps bind the functional imaging phenotypes across the whole brain, which is what you kind of see here from the matrix and the functional connectivity matrices across each individual, to patterns and dimensions of behavior that exist throughout the grouping, right? And so as a result, you now, instead of asking the question, how do many correlate to many, you kind of find a way to break it down into representations that capture the variance or the correlation structure within the data. And that can be then broken down into a smaller number of variables that you can then examine. So today the idea is to give you some intuition about these methods. What we won't be doing is, there'll be some math, but we won't be going too heavily into the math. But the idea is to provide some intuition about what these methods do and how to interpret them. I think that's probably a fair thing to do given the amount of time we have. So without further ado, I'll let Aurelie take the digital stage and we can go.
So hi everybody. So thank you, Mallar, for the introduction. And as Mallar mentioned, we are going to talk about 4 different multivariate techniques today. And I'm going to start with principal component analysis, or PCA. So first of all, what is the goal of PCA? The first goal is to reduce the dimensionality of a data set. So we can see that as reducing the size of a matrix in order to find new variables that we are going to call principal components. And why do we want to do PCA? So the first reason is that we want less complex information. So for example, if you have a matrix with 100 subjects and 1,000 features, it will be a huge matrix and it's really hard to find information about all the different information in this matrix. So we want to reduce the complexity. And we also want to allow better visualization. So again, if you have 1,000 features, it's definitely not possible to visualize these features at the same time. So with PCA, we are going to be able to do better visualization of your data. And finally, we want to reduce the size for 2 reasons. the first thing is that in this, for example, 1,000 features, it's likely that there will be features which are highly correlated. So it's maybe not useful to have all of them. And also we want to reduce the size in order to enable mathematical or analysis. So we want to reduce the computational time of any kind of analysis you want to do with your matrix. So first I'm going to go over all the different mathematical steps of PCA. And then I will try to explaining better with a visualization. So an example, when you want to do a PCA, the first thing to do is to standardize your matrix. So there is different ways of doing that. I'm just going to give you an example of a z-score. So here, for this example, we have 5 different subjects and 4 different features. And we can see that the values of these different features are pretty much in the same range. So between 1 and 8 for all of the different features. But for a minute, just assume that we had these values instead, where the values of F4 is much higher than the values of F3. And if you were running PCA on this matrix without any standardization, PCA will give much more importance into F4 information that it will give importance into F3 information. So we go back to this example, and in order to standardize, we are going to calculate the mean and the standard deviation for each features. And then we are going to apply this simple formula where the new value, the standardized one, will be equal to the original value minus the mean divided by the standard deviation. So if you apply that in all your values, you will obtain this standardized matrix. And just a quick note that in this case, the mean will be 0 for all the different features and the standard deviation will be 1. So then from this standardized matrix, we want to calculate the covariance matrix, which is defined like this, with the variance of each feature in the diagonal and the covariance of each pair of features in all the different other space in the matrix. So in order to calculate the variance and the covariance, we use these formulas, which are almost similar, with the variance being equal to the sum of the standardized value minus the mean square divided by n, which in this case will be 5 because we have 5 different subjects. So if you apply this formula for the variance and covariance, you will obtain your covariance matrix here. So now I'm going to introduce the eigenvalue and eigenvectors. So it's most of the time what is the most tricky about PCA to understand because the definition is a little bit hard to understand. So the definition says that the eigenvector is a non-zero vector that changes at most by a scalar factor that we call eigenvalue when there is a linear transformation applied. So I'm going to try with my example to explain a little bit more what is the eigenvector and eigenvalue. So here we are taking the covariance matrix that now I'm calling matrix A, and we are going to define this equation with the eigenvector being V and the eigenvalue being lambda. And from this equation, if you put everything, all the terms in one part of the equation, we obtain this equation instead. And here, A minus lambda I, with I being the identity matrix, which is defined as the matrix full of 0, except the diagonal, which is full of 1s. And basically, A minus lambda I here could be seen as this matrix for which we just subtract the lambda in the diagonal. In order to have this equation being true, there's 2 different options. There's either A minus lambda I equal to 0 or V equal to 0. But if you go back to the definition of the eigenvector, we know that it's a non-zero vector, so this is not possible. So the only solution will be that A minus lambda I, so the determinant of it will be equal to 0. So we can solve this equation. I'm skipping the steps for that. But basically here we will find that lambda is equal to 2.5. So if we go back to this equation, we can visualize it with the matrices instead. And since we know lambda equal to 2.5, we can now extract V1, V2, V3, and V4 values, which will be the values for the eigenvector 1. And we can do that again in order to find eigenvector 2, 3, and 4, for example. So here there is several things to say. The first is that we will obtain as many eigenvectors that we add features at the beginning. So we had 4 different features, so we obtain 4 different eigenvectors. In my first example, I was talking about a matrix with 1,000 features, so we will obtain 1,000 eigenvectors. Also, the variance explained by eigenvector 1, 2, 3, and 4 will be equal to 100% if you take them all. But something important in PCA is that eigenvector 1 will explain more variance than eigenvector 2, or that will explain more variance than eigenvector 3, etc. So basically, if you take the first eigenvectors, you will be able to explain a lot of variance in your data. So for example, eigenvector 1 and 2 here will explain, let's say, 80% of the variance of your entire matrix. So then the last step will be to transform the original matrix. So for that, we take the original standardized matrix and we multiply it by the top 2 eigenvectors, and we will obtain the transform data. So here we will obtain 5 different values for each eigenvector. So that will be the new coordinates of your 5 different subjects, 1, 2, 3, 4, 5, into the eigenvector axis 1 and the eigenvector axis 2. So now I'm going to try to explain it with a visual. So in this example, it's slightly different. So we have 2 features with the volume and the myelin, and we have 6 different subjects. So first you can see that the values of the volume and the myelin are not in the same range. So the first thing to do is to standardize the values, and then we can plot them. We don't really care here about like the specific coordinates. I just give an example. And then we want to center the data. So in order to do that, we are going to take the projection of each point into the volume axis, take the mean, and then take the projection of each point into the myelin axis, take the mean, and then the intersection of these 2 means will give you the center of your data. And now we are going to shift everything in order to match the center of your data with the origin of your. And now, if we want to define the PC1 axis, we are going to try to find a line crossing the origin, which will explain the most variance in these 6 different subjects. So we can try different lines, and basically we will find that this line will explain the most variance in these 6 different points. And this line is called PC1. So here I'm assuming that the slope is 0.33. So what that means is that if you move from 3 units of myelin and 1 unit of volume, you will obtain a vector in the direction of the PC1 axis. And by applying a simple Pythagorean theorem, you can find the size of this vector, which will be in this case 3.2. And I'm showing you that because now I can introduce the eigenvectors visually. So the eigenvector 1 will be the unit of PC1 axis, and we can relate this unit of PC1 to our original axis. So basically by doing this calculation with the 3.2 values that we calculated, we can know now that when we move from 1 unit in PC1 axis, we move from 0.31 unit of volume and 0.94 unit of myelin. So now we are going to define the PC2 axis, which is always the orthogonal line of PC1. So in this case, since it's a 2D example, that will be basically the perpendicular line of PC1. I'm just going to give you a quick note that most researchers consider that PCA is useful only if the correlation is higher than 0.3. So it's easily understandable when you visualize it. So if, for example, I shift a little bit my data, and now we have a very low correlation, let's say 0.1, you can see that PC1 axis and PC2 axis are almost similar to the myelin and volume axis. So there is not really any goal to use PCA in this case, because PC1 and PC2 axis will not explain better the data than your original axis. So now we are going to flip the axis for a more intuitive visualization. And the last step will be to calculate the variance explained by each PC. And for that, we use these formulas where the variation of PC1 is defined as the sum of the square of the distances for PC1 divided by n-1. And here the distances for PC1 will be the projection of each point into the PC1 axis, so these distances. And then we do the same for the distances of PC2, so the projection into the PC2 axis. And now I'm going to assume that PC1 variance is 15 and PC2 variance is 3. So that will allow us to calculate the variance explained by each PC. So for PC1, we will find that PC1 explains 83% of the variance, while PC2 explains 17% of the variance. So I'm just going to finish with some benefits and drawbacks of PCA. So the main benefits, I talked about it before, so is that the correlated features are removed, which improves the performance of the algorithm, reduce the overfitting of the model, and improves the visualization. But there is also a few drawbacks. So the first one is that PCA assumes that the principal components are a linear combination of the original features. But if you look at this graph here, you can see that the data are not really following any linear relationship. So in this case, PCA is going to struggle to make sense of the data. Also, PCA assumes that the high variance axes are principal components, but the low variance axes are treated as noise, which is one of the main properties of PCA. But depending on your data and your research question, maybe the low variance axis could be of interest too. So in this case, it's maybe not the best option to use PCA. And finally, PCA assumes that the principal components are orthogonal. But if you look at this example here, if this line would be PC1, the perpendicular line in this direction will be PC2, but you can see that it doesn't really fit well your data also. So in this case, it's probably better to use another technique than PCA. So I would like to think, oh, sorry, I have a few references for you. So if you want to run PCA, either R or Python, it's just a few links in order to help you for your code. And with that, I would like to thank you for your attention, and I will let Elisa continue.
Great. Thanks so much, Aurélie. Do you have any questions while we're kind of swapping out speakers? Certainly you can chat me questions, or if you want to ask them later, you can hold them to later as well. But that was a great overview of the technique. Thanks.
Thank you.
Anyone? Nothing? No hands up? All right. So we're gonna, in the interest of staying on time, we're gonna keep moving. When would you favor? I don't even know what your. The question is, when would you favor TSNE over PCA? Can you spell out TSNE, please? Okay. Well. Okay. All right. Well, maybe we can talk about that later. I think the question really is okay, so there's some really good questions. So I'm not sure what TSNE is. I've never used it before. T distributed stochastic neighbor embedding. That's not a technique I'm familiar with. It sounds very cool though. So unfortunately, I don't know that we have any answers for you there. Vlad Fonov is asking a question of how can you run statistical tests, i.e. t-tests, on PC vectors. So Aurélie, do you have any answers there? So every subject will have a weighting.
Coordinates, or
Yeah.
like weighting on each PC. So it's just like basic test after that. I think you just take the new values and then you can then like take your statistics and then you can relate it with your original axis, and I try to show with the visualization. So yeah, I don't think there's much more.
I suspect, Vlad, that is a question you know the answer to. Anyhow, what about multiple comparisons? Well, then you would use multiple. I mean, wouldn't you just correct for multiple comparisons over the number of principal components you did statistical tests on? Wouldn't that? That's how I've done it in the past, but I don't know that if there's an issue with that. I don't think there is. All right, I'm going to take the silence as agreement, I guess, tacit agreement. And I will hand over the reins to Elisa Guma. And so we'll take questions again after this talk. Thanks.
Great. Thanks, Mallar, for the intro, and to Aurélie for introducing a lot of the key concepts that are going to be carried out through these different methods. So I'll be talking about PLS today. So PLS is a multivariate statistical technique which was first introduced to the neuroimaging community by Randy McIntosh and colleagues with the goal of extracting commonalities between brain measures and behavior. So he identified 3 different variants, but we'll be focusing on a correlation variant today, which is the behavior PLS, which aims to analyze information common to brain and behavior. So here's a nice representation of the entire workflow from a figure by Krishner and colleagues, which shows you how you get from your inputs to your results, which are defined as latent variables. And this shows you how to get one latent variable, but PLS will give you multiple. And I'll be defining all these terms as I go. So your first input matrix is going to be your brain matrix or your X matrix. So here you'll have your participants as rows and then your brain information in the columns. And in this example, they used voxel-wise deformation-based morphometry measures, but you can imagine putting something else in there. So it could be metrics derived from diffusion-weighted imaging, such as fractional anisotropy. It could be microstructural metrics, such as T1 over T2, or vertex-wise cortical thickness or surface-based metrics. If you want to go a level of granularity above, you can use structure volumes generated from an atlas-based segmentation like maggot brain, or alternatively, you can even use outputs from a different data dimensionality reduction techniques such as PCA, which Aurélie has just explained, NMF, which Ray will go over shortly, or ICA4, just as a few examples. Your second matrix is going to be your Y matrix or your behavioral demographics matrix. So here for the same subjects, you're going to have something like a clinical or cognitive score, which is what Krishner and colleagues use. They had symptom dimensions and cognitive scores for individuals with schizophrenia. But you can imagine having behavioral data from other tests, such as a series of memory tasks. And this could be from humans or mice or rats. Or monkeys, whatever you're working with. You'll also be inputting your demographics information, so sex, age, socioeconomic status, or you can use data gathered from postmortem work, such as histology or gene expression data. So your next step will be to normalize your matrices, which is typically done by z-scoring them, and create a correlation matrix. And then on this correlation matrix or brain behavior matrix, you're going to run singular value decomposition. So SVD, singular value decomposition, is simply a data reduction technique. It allows you to take high dimensional data and distill it down into some key features to pull out important information. So in this case, it's going to break your matrix down, so your Z matrix, into 3 separate matrices from which we can get our latent variables. So in this setup, Z is our correlation matrix. Then we would get U, which is the left singular vector matrix. So the columns are important here. You get V transpose, which is the right singular vector matrix. And so essentially, these matrices weight the original variables in your X and Y matrices in such a way that they maximally covary. And the third matrix, sigma, is a matrix of singular values along the diagonal. So everything else in this matrix is 0. And these singular values get progressively smaller as you go down the columns. So the first column will be more important, let's say, than the second, explaining the covariance between these 2 matrices. And in this case, U1, sigma 1, and V1 would define your first latent variable, and then U2, sigma 2, V2, your second, and so on. And just as a note, the dimensions of your V transpose matrix are as wide as your clinical and cognitive, the number of clinical cognitive inputs you had. So that will define the maximum number of latent variables that you'll get from this analysis. So just to so just to define latent variables again, they're orthogonal to each other and they're linear combinations of brain behavior that maximally covary. And just to bring it back to our example, so you can get a brain score, which you derive from the U matrix. This is a projection of the weighted pattern U to the original data from your brain matrix X. And in this case, you can visualize it as a DBM pattern if that was your input. You get a singular value from the sigma matrix, which captures the proportion of covariance explained by the latent variable that you're looking at. And finally, you get a behavior score, which is a projection of the weighted pattern from your V matrix to the original individual data from the Y matrix. And so in this case, again, we're visualizing hypothetical results for the first latent variable, but your second latent variable would have a different pattern. So now that we have our latent variables, we want to assess their significance. And we do this using permutation testing. So essentially we start by reshuffling the rows of your brain matrix X, which removes any existing brain behavior correlation. And we then continue on with the same steps outlined before. So the Y matrix stays intact, we generate brain behavior correlation, run SVD, and obtain our singular values. Then we repeat this, and we do it again 1,000 to 10,000 times, which allows us to generate a distribution of singular values under the null hypothesis that there is no relationship between brain and behavior. We can then compare our original brain behavior relationship generated by our actual PLS to the null distribution of generated by this permutation testing and derive p-values. So in addition to assessing the significance of our latent variables, we also want to understand the contribution of each input variable, so voxels or behavioral measures, to the given latent variables. And we do this via bootstrap resampling. So here we're going to reshuffle both the X and the Y matrix, and then create a resampled correlation matrix, run SVD, and repeat again 1,000 to 10,000 times, depending on what you're feeling, I guess, or how much your computer can handle. So again, this would generate a sampling distribution for each individual weight in the singular vectors. And then you can get a bootstrap ratio calculated at each voxel as the ratio of its singular vector weight to its bootstrap estimated error. So you can think of, let's say, voxels with large bootstrap ratios contributing a lot to the brain pattern. Same with the behavior. Okay, so now I'll just contextualize, sorry for my voice, with results from Krishner's study. So they used PLS to assess the clinical anatomical signature of schizophrenia. So from their PLS, they detected 3 statistically significant latent variables, and they chose to visualize the results for the first latent variable here, which accounted for 27.5 of the covariance between their MRI and their clinical cognitive data. So here we have the clinical cognitive scores for the first latent variable, and here we have the deformation-based pattern for the first latent variable. And essentially what you could see is that patients who display this deformation pattern tend to have higher scores on symptom dimensions, particularly the SANS, which is the negative symptoms, and they also tend to have lower cognitive scores. So that's an interesting way to integrate all these clinical and cognitive scores with one brain pattern. I've also used PLS in my PhD. So I collected, or we collected data on mice who had been prenatally exposed to maternal immune activation relative to controls, and collected a number of different behavioral measures on them as well. So instead of running single tests between brain and, let's say, distance traveled in an open field, we wanted to assess a kind of bigger picture of brain behavior covariation. So what we were able to pull out is that regions in red were associated with a pattern of increased or decreased locomotion, but better performance in social behaviors and sensory motor gating, whereas regions in blue were associated with the opposite pattern. So poor performance in social behavior and sensory motor gating and increased locomotion. And just to sum up, some advantages of this technique are that it can handle large data sets, which are typical to neuroimaging research. So you can throw your voxel-wise DBM maps at it. And it's also a more elegant way to explain the relationship between multiple types of data at the subject level. Some disadvantages are that it can't handle missing data, so you may have to perform some sort of data imputation technique if you have some missing information. It assumes a linear relationship, which again may or may not be a disadvantage, and sometimes interpreting what your latent variables mean can be challenging. Just with that, I'd like to point you to a couple of resources. So Stephanie Tulo in our lab has written a wiki on how to run this in MATLAB. Ross Markello from Bratislav Misic's group has implemented PLS in Python, which I hear is great and very easy to use. And here are a few papers as well that kind of either use PLS and explain it really well, or just talk about PLS in general. So with that, I'd like to thank you for your attention and take any questions if you have them.
Great. Thanks so much, Elisa. So we're going to maybe use this time to swap out presentations so that Lani can put hers up. There was one question from Nadia, which was, I won't find it, but by PCA decomposition, so she's talking about the feature reduction step that you mentioned, Elisa. You're not inferring statistics from a sample to a population. You're just creating new features, correct? And yes, that's correct. It's if you want to reduce the dimensionality, if you have a big feature space to begin with. It's just a one way of potentially pre-processing your data. So you could, the question is, could you bootstrap your input feature in a PCA test to test the stability of the PCA components? For sure. You can do whatever you want on that end, I think, to test stability. That was just an example of how to reduce dimensionality if need be at the beginning. Anyone else have any questions, comments before we move on? All right, Lani Cupo, it's all yours.
Hello, everyone. My name is Lani Cupo, and I'll be providing kind of a high-level overview of canonical correlation analysis, as it does come up in some CIC lectures and many papers these days. A lot of the work for this presentation comes from this paper by Wong et al. In Neuroimage 2020. So if you want to do a bit of a deeper dive, I highly recommend this, as well as some other references that I include later on. So in many ways, CCA can be seen as an extension of PCA, which Aurelie provided such a nice overview of, except instead of a single input matrix, we have two different sets of variables that we're trying to relate to each other as well. So for example, if we have a first variate or set of variables that we'll say that we have the volumes in it. So variable 1 could be hippocampal volumes, 2 could be striatum, etc. And a second variate, y, where we have, let's say, behavioral metrics. What CCA is really trying to do is find linear combinations between two random variables to maximize the correlation between the combined variables as well. And it doesn't have to be volumes and behaviors. It could be genetics. It could be groups of voxels, anything like that. You'll notice that we have the same number of participants for both X and Y, but P and Q, or the number of variables, can differ. It's just important that N is greater than the maximum of P or Q. We want more observations than we have features or variables. So first, I'm going to start with kind of a high-level conceptual overview. Given our variates, X and Y, that comprise sets of variables that we discussed before, we submit them in parallel to singular value decomposition. So that gives us what we call canonical vectors, A and B. And then we take the weighted sum of these variables times an element in the vector, and we get what is called a canonical variate, U and V. And a pair of these canonical variates is known as a mode. So then we correlate U and V for all of the subjects, and that is the canonical correlation, which is the linear correspondence between the two canonical variates, and it's the main performance metric of this technique. So that was a lot of information. Before you ask questions, let me give kind of a more illustrative example. So say we have 30 different participants, And we have demographic data for them, p equals 4, and academic data, q equals 5. And we want to know the relationship between the demographic data and the academics. So in the demographics, we have their age, gender, their house at Hogwarts, and their hometown. And then we have their marks on 5 different classes, and we want to know the relationship between them. So we can take our first subject, Harry Potter, and with singular value decomposition, we can compute U and V. And this becomes the first subject's correlation for the first mode. And then we can repeat this. And again, like PCA, we can produce multiple modes that are orthogonal to each other. And they are the variance that was not explained by the previous mode. So we can do this again for Harry Potter until we have 4 modes. And then we do that for our next subject and all of the subjects until we have this matrix that has our subjects and their results for the modes. The number of modes is less than or equal to the minimum of P or Q, which is why we have 4 here, because we had 4 initial variables within our X matrix. So then we take our subjects and their first mode and we plot that, and we get the canonical correlation between U and V, and we do this for all of our modes. And now that we have all these modes, we have to know which of them are actually significant or meaningful. So the most common way of doing this is a permutation-based non-parametric testing. So we take that X matrix and we shuffle the rows, kind of as Elisa described as well, and we get X hat. And then we can recalculate the permutations between X hat and Y. Notice that we don't do anything to Y. And we can create a distribution of these correlations. So we reshuffle X, recalculate the correlations, and add them to the distribution. And we can do this 10,000 times or so until we get our null distribution. And then we can compare the real correlation from each of the modes to this null distribution. And that's how we get our p-values of significance to see which of the modes are actually meaningful. So I've been discussing the main type of CCA. However, there's different variants. So in case n is not greater than the maximum of P or Q, you can either first do a data reduction step and then CCA, or there's options for sparse CCA as well. If you think that the relationship between X and Y is nonlinear, then there's different options for doing a technique that will allow for that. And if you have more than 2 variates, or X, Y, and Z, then you can also do a multi-set CCA to find the relationship among all of them. So finally, I'd like to take you through a bit of more applicable neuroscience examples. This is from Smith et al. In Nature Neuroscience 2015. And their goal was to relate one matrix that had functional connectivity data with another that had behavioral and demographics. Their data came from the Human Connectome project, and they had resting state fMRI data, as well as behaviors and demographics for 461 participants. And their first steps was the pre-processing. Actually, their pipeline is outlined quite nicely in the Wong et al. Paper. So first they constructed these network functional connectivity matrices. And then these, as well as the Z-scored behavioral tasks, were submitted to PCA. And this was to ensure that they had a smaller number of variables. So they took the top 100 components for both of these matrices, and that was their input to the CCA. So X had the first 100 variables and Y as well. So only the first mode that they found was actually significant. So that's what they examined. And on the left, you can see the set of phenotypic variables that were associated with mode 1, where the blue variables are included in the CCA, and the gray variables were correlated with the results post hoc. The vertical position is related to how strongly each variable correlated with the CCA mode, and the font size indicates how much of that variance was explained by CCA. So up in this right-hand corner, what the trend you're looking at is the correlation from mode 1 between the connectome on the x-axis and the phenotypes on the y-axis with 1 dot per subject. And as an example, they visualized the positive correlation for fluid intelligence, which is the second variable up here, in different colors. And then finally, for the first 20 modes, they also show the variance explained for each, the connectome up here and the phenotype on the bottom. And the gray shaded area represents the 5th to 95th percentile for the null distribution. So this is how you can really see that only mode 1 was significant for them. Then this visualization represents the 30 connections that were most strongly correlated with mode 1. So the pattern that maps onto the phenotypes in the last one. And the red indicates a stronger connection and blue a weaker connection. So their conclusion was that they found one mode linking connectome and phenotype, and they said that positive traits such as higher education more strongly correlate with this pattern, and negative traits negatively correlate with it. So there are some limitations to CCA, for example, and as we've said, must be greater than the maximum of P or Q, which is not true for PLS. So you may have to do a data reduction technique, and sometimes that can complicate the interpretation. While not required, normal distributions in the data are desirable, so that's something to keep in mind. And this isn't a limitation, but whereas CCA maximizes correlation, PLS maximizes covariance, which is one of the biggest differences between them. So in conclusion, CCA represents a powerful tool to reveal underlying patterns between two or more sets of variables. And if you're interested in reading more about the process or the math or implementing it yourself, I highly recommend these references. Thank you.
Thanks so much, Lani. So here's a really great question that I figured was going to come up, and maybe Elisa and Lani could tackle this if you wanted to. When would you use PLS versus CCA? It seems that the input in both cases can be similar, and the output seems also similar, latent variables versus modes. So I'm wondering how I should make my choice. So excellent question. And go for it, Lani or Elisa, if you'd like.
Yeah, so I guess one of the major things that came up for me was if you are looking at like voxels, for example, and you know that you're going to have way more variables than you have participants, that might, unless you want to do first PCA as they did in the Smith paper, it might make more sense to do PLS off the bat. Because it can get a little bit complex to interpret like latent variables of latent variables.
So yeah, I think the dimensionality of the data really matters a lot. And do you want to look at correlation or covariance as a metric of the relationship between your variables too? I think that's really important as well, because I tell you different things about the data as well. Ray, if you want to jump on and share your slides, go for it. Great. And last but certainly not least is Raihaan Patel, who will be talking about non-negative matrix factorization. And we have a question. Sorry, before we start, is there a min-max number of variables you should include in PLS relative to the end? I don't know of, I might be wrong here, but Elisa, you probably looked into this more than I have, but I don't know of a rule of thumb there.
No, I haven't come across anything specific. I guess it kind of depends. Like the more behavioral data you have, the more latent variables you have, so the less kind of shared covariance would be allotted to each latent variable. But I usually tend to choose variables that I know are informative. Like from my behavioral data, I can get a ton of different metrics per mouse, but I'll choose like 4 to 5 per behavioral test because that is as much as is going to be informative. But I don't think there's a hard and fast rule on how many you should have.
Yeah. By consideration, forget about the PLS, but make the same case for the CCA, the Smith paper that Lani mentioned before. They do all sorts of pre-processing on both their MRI and demographic behavioral data. So like she was mentioning, you have this issue of latent variables and latent variables if you decide to go too big. But I think, one thing we struggle with is, and it's a question of exploration and comfort, I think, is the trade-off between, I guess, interpretability versus data exploration. And I think that will then in turn inform the end of your inputs too, probably, right? Because at some point you include all the things, but then you have to interpret all the things, and that becomes borderline impossible too, right? So, defining your question is really important.
Yeah, if you have like a strong hypothesis, it can make a lot of sense to reduce what you include just because you're going to find something that's a little bit more understandable off the bat.
Yeah, Vlad, I believe the question is, Do you need to pre-process your data in a PLS? And I think basically in all these techniques, we need to pre-process the data so that we don't give kind of some unfair weighting to one metric over another. So I think that's a key. You know, the nature of the normalization is, you know, people sometimes debate that, but certainly you need to. Yeah, NMF does have some additional normalization class to be seen soon. Agreed. All right, which is a perfect introduction to Raihaan's talk. So go for it, Ray.
Cool. So my name is Raihaan, and I'm going to be talking to you about non-negative matrix factorization, or NMF, the final three-letter acronym for today, in the context of neuroimaging analysis. So today, I want to go over what NMF is. I want to give you a couple examples of how it's used in neuroimaging, such as those figures on the right. So from this, I want you to be able to understand generally what's happening if you come across NMF in a paper, as well as if you have the type of data that it could be used for, just to have a general idea of what benefits it may offer you. And then I'll briefly touch on how NMF works. So if you were to run this algorithm, what's happening in the background with your data between input and output. So to start, what is non-negative matrix factorization? It's a matrix factorization technique which decomposes an input matrix into two matrices. So a common syntax you'll see is shown here. The input matrix X is decomposed into two matrices W and H. And as denoted by this symbol here, the multiplication of the two output matrices, W and H, is an approximation of your original input data. So by that, it's aiming to capture the major patterns of your data. And then another consideration, as suggested by the name, the input as well as the outputs are all greater than or equal to 0. They're all non-negative. So that's important when it comes to the interpretability of the outputs. And in the context of neuroimaging analyses, if you think of structural MRI data or diffusion MRI data in particular, though that's usually data types, whether it's cortical thickness or volume or a T1 weighted value, or maybe something from diffusion like fractional anisotropy. Those values themselves are all non-negative as well. So in that way, it's a sort of logical fit for some of the data that we're used to working with, and that can help in how it's interpreted. So the way that looks visually is shown here. On the left, you have some sort of input matrix, and then you have the two outputs on the right, the component and the weight matrix. So these are all matrix-based techniques. So if you're thinking of working with this, you're going to have to end up building some sort of input matrix with your data. And then an important distinction in comparison to the techniques we've heard is the approximation part here. So while PCA or CCA actually aim to capture all or much more, at least, of the patterns in your data, NMF is typically more of an approximative technique. So it's going to give you the high-level patterns, but the degree to which it perfectly reconstructs your input is going to be much different and much less than some of the techniques you've seen before. And so the reason for doing all this, why bother breaking apart your input into two different outputs, is to give you a parts-based representation with additive components. And I often think of NMF as a variance detector. There was a paper that, or a few papers that I've read, where in the discussion section, when talking about the benefits and trade-offs, they essentially describe it as a way to describe or to identify where interesting stuff is happening in your input data. And that's what I think about a lot when considering whether or not it's worth trying to use. So in this visualization, each column of this matrix is a component, and it has an associated set of weightings, which is each row here. And those aim to reconstruct your data. The number of components, so in this case, the number of columns in this matrix, a variable often referred to as K, maybe also known as the granularity, that's selected by the user. So that's selected by you. So that's another important distinction compared to the techniques you've seen here. When you run a PLS or a CCA, you get some level of significance for each of the output components, but you don't have to say at the start, give me 5 components. Whereas in NMF, you do have to do that. And I'll talk a bit about ways you can make that selection later on. And then your input matrix X, as I mentioned, it's reconstructed as a linear combination of the component and weight matrices. So when we talk about a parts-based representation or additive components, I just want to show a bit of matrix multiplication here. So on the left, I've highlighted a certain element in your input matrix. And then on the bottom, I'm showing how it's reconstructed or how it's modeled based on the two outputs. So you can see following this equation that it's reconstructed as some sort of weighting of each of the three components. And that's where the interpretation of the parts-based or additive component representation comes into play. So now I have a couple examples of how it's used in neuroimaging. So in this figure here from Nassar et al. In cerebral cortex, you again have your three matrices. On the left, you have your input matrix, and then we have component and weight matrices. In this case, what the authors did was they took voxel-wise volumetric data and applied NMF to that. So in their input matrix on the left, each row is a voxel, and each column is a given subject's voxel-wise volumetric data across the cortical surface. And then in their component matrix, they end up with, for every voxel, a component score. So you can map that back to your surface, and you can say where does a given component exist. And you also end up with a distributed set of weightings for each of your subjects, for each of your networks or components. So in this case, they're breaking apart their volume data into a component where you have a measure for each subject describing the volumetric properties of that specific area. So I think of this kind of in comparison to atlas-based cortical thickness or volumetric techniques. Say you ran cortical thickness pipeline and you get a number of measurements across the cortex. What you might do is you might apply an a priori anatomical atlas and then take the mean cortical thickness or the mean volume in a given region. So instead, what's happening here is NMF is kind of defining your regions for you. And then again, you're looking at a measure for each subject within each of those identified components or regions to describe some sort of variability. And then, of course, just like you would with an atlas-based approach, you can take those subject-wise values and run some sort of statistics on them. Say you could relate them to age or some sort of behavioral test. Another way you could do this that I worked on in the hippocampus using Human Connectome Project is to integrate multimodal data. So because NMF is good at modeling covariance across metrics, here what I did was I included T1/T2, mean diffusivity, as well as fractional anisotropy data to derive 4 components of the hippocampus. So this describes group level patterns as well as individual level weightings as well. So I mentioned choosing the number of components or K. So the way that's typically done is some sort of trade-off between the reconstruction error in blue or the accuracy, as well as some level of spatial stability, where stability is describing how the spatial components are represented across all splits of your subjects. So what you want to avoid is a set of components that are just driven by some sort of outlier subset. You want components that are relatively identifiable across all your data. And then I'll briefly touch on how are the two outputs computed. So if you run NMF, what's happening in the background. The first step is to initialize them. So they're matrices, and you can initialize every value in those matrices to be random or to be 0 to start. What's actually more commonly done in practice is to use a singular value decomposition. This helps promote sparsity in your outputs to make sure your components don't overlap. It also helps you capture some of the main patterns already, so it gives you a bit of a head start. And then you compare the error, so you look at how good is your approximation. This is an example of a typical objective function or cost function. So this is identifying the difference between the original input and the reconstructed input. And you have some sort of stopping criteria. So you want to ask how good is this approximation. So in this pseudocode here, it's comparing the error at a given iteration to the initial error. And what typically happens is there's some sort of threshold that says when the error at a given iteration is 0.1% of the initial error, then it is good enough and we've captured enough patterns, and then you can stop there, although that can vary, but that's the general idea. And then of course, if that criteria is not met, you repeat and you update W or H until the stopping criteria is satisfied. So some of the benefits and limitations. So as I mentioned, it models shared covariance across input data. So it's a great way to capture, it's a great way to capitalize on multiple input data types. It gives you an additive parts-based representation, which can help with your interpretation. It has different variants, so you can adapt that for continuous versus discrete components if you want continuous components versus sort of a hard clustering. And it gives you data-driven identification of spatial components. That's often touted as a benefit, but I think that also summarizes the limitations as well. So it identifies regions specific to your data, but that also means it's up to you to decide the relevance of those spatial components. And then a part of that is, of course, that you have to define the number of components as well. And I'll stop there and I can take any questions.
There you go. So it's just shy of noon. So great talks by all of you, first of all. Thanks so much. That was very well done. And thanks for preparing all that stuff. Any questions about non-negative matrix factorization or any of the techniques that were presented today? Vlad is asking, you mentioned pre-processing for NMF.
Right. So sort of like how Aurelie mentioned, she had some columns that were different data types on different scales. So if one is 400 and then one column is like 0.1, because all the algorithm is doing is trying to minimize error, it'll benefit from paying more attention to one column than the other. So if you have just cortical thickness data, that's probably not an issue. I would guess that across all vertices, the data is on relatively the same scale. But if you were to do something like combine T1 over T2 data, which has a range of 1 to 2, and mean diffusivity data, which is like 0.000 something, you would need to do a Z-scoring or something as well, just to make sure that all the data is on the same scale.
Vlad, does that answer your question? In the meantime, what would be the difference between machine learning techniques and the techniques presented today? I'm knowledgeable in either. Is that the input given to the techniques presented today are more theory-driven and need to be chosen carefully? So I'll let Ray answer that. I'm sure Ray has a very good answer to that. Sounds like a very excellent thesis defense question.
I think the stuff always needs to be chosen carefully. So I guess you could say most of the stuff here is unsupervised in some way. So if you choose different machine learning techniques, that could change. But I think all the techniques here are more suited, or at least assume some sort of linearity. So if you get into more complicated machine learning techniques, that can go away. So then that can be desirable or not desirable. But if in terms of like choosing what to use, I think the stuff here is pretty appropriate for detecting patterns in your data. But if you're thinking of like prediction, so I'm thinking of comparing NMF to a support vector machine or something. Matrix factorization will detect patterns and say, here are some components and here's what stuff is doing. But the support vector machine is more suited to make a prediction based on the input. So that goes back again to the unsupervised supervised type approach as well. And then I guess you could put if you were thinking of like neural networks or deep neural nets or something, that would be in the same category. So in those, you have your input and then you have some sort of predicted output. So maybe you're trying to learn, is this person a patient or not? Whereas in these techniques, it's just saying, show me the patterns, and then I'm going to do stuff with them afterwards. Does that make sense?
You got a big thank you. There you go. And Vlad asks how to deal with negative data points.
So for NMF, as the name suggests, it has to be 0 or greater. So you could shift or do some sort of shifting that would be involved in your pre-processing. But if you have negative data points as well, that moves away from like the novelty or sort of the applicability of NMF. But if it was like some of your data is negative and some of it isn't, then I think you could shift the data as long as you maintain the distribution. And that would be okay. I've done that before as well.
Anything else? All right. Thanks everyone for your attention. Thanks for showing up. Great talks, guys. Very impressive. I hope I hope the audience got a lot out of this. And yes, Stefan, the recordings are posted every week on YouTube, kind of subsequent to the talk. So thanks, folks. Have a great lunch, and we'll see you all next week. Take care. Bye.